talk in our morning session. I'm very excited to introduce Robert Cass, who is a professor in the statistics department at Carnegie Mellon University. He's known for his great work in Bayesian statistics, as well as applications in neuroscience.
Thank you. So, a little over 15 years ago, when I first got interested in neuroscience, I had multiple conversations with Emory Brown here, where we commiserated about our common experience of reading multiple articles in neuroscience and kind of had the same reaction where we would see lots of data analyses by people who are clearly very sophisticated in math and in computation, and yet we would come away from it feeling that we would have approached the problem differently and we would have presented the methods differently. And in this process, we thought a lot about this, and I ended up asking a whole bunch of people, whoever I could find, really, what was the difference? In other words, what's the difference between someone who's trained in statistics and someone who's not? Or to put it differently, what does statistics bring to the research table? And I asked people, many of them in the room. I remember asking Steve and Peter and Rob, Mike, I believe you may not remember, but I do. Trying to get opinions about how you could define this, and I thought it was useful to try to distill it down to a couple of very short statements. Useful, really, so that we could concentrate on these things when we teach, especially. So what I'm going to start with is an answer to that question, and it's the question is one way to put it is what is statistical thinking, and I want to give what I think is a consensus answer. I want to say, by the way, I think everybody who I would have asked, and I certainly read a lot of what Andrew was writing as well, everybody would choose different words, I think, but I'm hoping that the spirit of what I'm saying is really a consensus. And I'm going to give I'm going to follow that with a very simple Bayesian example for pedagogical purposes, really to show how this is something we can get across in the most elementary undergraduate courses. I want to add a kind of a pitch for statistical work in networks within neuroscience. That's going to actually come a little earlier, primarily, and I'll say something, just provide like one small step forward in that domain in something that we call FDR regression, false discovery rate regression. And at the end, we'll comment on something that I've only recently really started to follow at all, reproducibility, replication and reproducibility. And all of this really touches very closely on issues that both June and Andrew have already discussed. I think it actually meshes very well with what they've done. So in mixed audiences of this type who don't necessarily have a lot of familiarity with neuroscience, I always try to start with kind of just a little bit of neuroscience background, and I found very useful for this purpose a graphic that came out of a textbook which I find illustrates really quite nicely something that probably most of you already know, which is the brain is located in the head. And you probably also will recall that neurons communicate primarily through rapid electrical discharges known as action potentials. We also call them spikes. They take place over about 1 millisecond, and on a behavioral time scale of several hundred milliseconds, the time it takes you to move your hand, for example, it's essentially discrete points in time. And this is called a spike train, and a lot of the stuff that I've worked on, and also a lot that Emory's worked on, has involved the analysis of spike train data. The natural statistical framework for this is point processes. The data come typically as binary time series, sequences of zeros and ones, let's say in 1 millisecond time bin, a 1 if there's a spike and a 0 if there's not. But the simplest textbook answer to the question, how do neurons carry information, was established in a bunch of work now quite a long time ago, and it's still a dominant answer in the field. It's that the neuron responds to a relevant stimulus or contributes to the production of an action by increasing its firing rate. And this is, again, a textbook cartoon of what's done in real experiments where one neuron is recorded from the brain of an animal, here a monkey, in this case from primary visual cortex called V1. So it's again one neuron. He's looking at what's called a fixation point. He's got to hold his eyes there. in order to get rewarded, he cannot move his eyes. A stimulus comes on. It's actually a light bar that's moved across a particular location on a screen in front of him. This is called a receptive field. When it's moved across that field, the neuron starts to fire very rapidly. If it's shown somewhere else, it fires much more slowly. And I have a little film clip to show you. This was done 50 years ago, and all they're doing is they're moving a wand of light across the screen, and you'll hear the neuron firing when the wand goes across the receptive field. So there's a receptive field, and I like this because it's a very vivid visual image of what's going on, and also you don't need any statistics. But these days things are much more complicated. On the left is a picture of the way data of that type is collected, a single electrode going into a region of the brain near one of the neurons that's now being recorded. These days there's a variety of multi-electrode arrays. This is one of the most commonly used, the Utah array here, shown next to a US penny. And it allows much richer data collection. This is a slide that was made by a student at Carnegie Mellon, former student. I was on his thesis committee, he's a computer science student actually, but then he was a postdoc with me for about a year and a half. And what's shown is now, you're going to see down here a bunch of spike trains from 128 neurons recorded at the same time on one of those arrays, again from primary visual cortex V1. And up here are circles. Each circle corresponds to the receptive field for one of the neurons, and when it's red, that neuron is firing rapidly. When it's black, it's firing less rapidly. The monkey is actually anesthetized. His eye is propped open, and he's seeing this movie in front of him. And the main point is really just, I'm showing you that we have a rich data set, and it's a much more complicated statistical problem to try to figure out how does this set of neurons encode the information in the stimulus. There's now been a lot of work, continual work on trying to record more and more neurons simultaneously. Actually, the rate's been shown to be the rate at which the number of neurons that can be recorded at the same time has been growing exponentially fast. There's a variety of technologies for this. And it's being combined with potentially somewhat complicated behavior. Here, there's a couple dozen neurons being recorded from a rat who's executing a reward-related behavior, but now this number is going up to hundreds, and soon it will be thousands or more. I also wanted to mention there's a very exciting technology just from a few years ago, optogenetics, in which you can insert into a certain particular neurons ion channels that are sensitive to a particular frequency of light, and then you can drive those neurons to fire and look at what the consequence is for either behavior or for downstream neurons, and this gives you actually causal relationships. So all of this is just to say that there are great opportunities here going forward to answer the question, opportunities for the 21st century in statistics. Standard big data analysis and big data issues are arising, some of which have already been described. But here we are grappling with model complexity while trying to assess the reliability and relevance of the results. A lot of this involves networks of one form or another, dynamic networks, covariates to deal with the behavior and so forth, and of course always assessment of uncertainty. So what about statistical thinking? All right. In order to connect with that, I want to go back to what I was mentioning earlier and give you the answer that is in this paper that I wrote with Emory. Here's the two statements that we think capture the, the fundamental tenets of the statistical paradigm. It's not comprehensive, but I hope you'll agree that it does a reasonable job in summarizing briefly something we can continue to try to emphasize as we talk about the role of statistics. First, statistical models are used to express knowledge and uncertainty about a signal in the presence of noise via inductive reasoning. And second, statistical methods can be analyzed to determine how well they're likely to perform. You saw both of these discussed in the two talks earlier. I'm not claiming it covers all of statistics. There's exploratory analysis. There's a lot of computation. There's a lot of algorithmic issues. There's there's also, I should say, data collection, which is really important. It's come up in the discussion, and I'll come back to that at the end. But I do think when we're trying to say what's the difference, when someone has statistical training, this goes a long way toward capturing the unique contribution of the discipline. Because, again, the people who we were complaining about in the literature were not starting with a statistical model. And by the way, when I say model, I don't mean parametric model. All I mean is there's some probabilistic notion. It can be very, very general. Also, the first item is far from obvious. This is the reason people don't gravitate to it automatically. It's something where training is actually important. And the second one involves principles, and every field has their principles. This is something that is extremely important to us and should be taught, in my view, to everyone at every level of statistics. We need to emphasize the principles, and the second item is really oriented toward that. To emphasize or to illustrate how I believe we can do this, even in the most basic statistics course, if you go to the most common measurement criterion, I should say, for performance, mean squared error, you can illustrate this without doing math. You can show people what high bias and low variance is, low bias, high variance, high bias, high variance, or low bias, low bias, low variance. You can talk about these things. Actually, I've done this many times with neurobiology students who are taking their first course in statistics, and I think it's pretty effective. Also, when we introduce confidence intervals, I'm personally much less worried about the interpretation of those, but I do want them to understand that they should cover the estimated quantity with about the right probability. And here's my simple example. Again, I like this example, I should say, because it's elementary. It's actually novel in statistics, I believe, but it's so elementary that we couldn't possibly have published it in a statistics journal. And again, it's something that can be explained to elementary students in neurobiology or neuroscience. So it comes from an experiment that was done, and I happened to be in the room when results were reported by a graduate student working with one of my colleagues. And it concerns the attenuation of correlation. And what my colleague was concerned about was a phenomenon that had been reported in the literature by another lab, which involved selectivity for serial order among neurons from the supplementary eye field. Supplementary eye field is just forward of the motor area, and it feeds into the area that controls eye movement. And it wasn't clear exactly what that area does. And this particular lab said, well, if you train a monkey to move his eyes to three places, three different images on a screen sequentially, some neurons will fire for that third image more than they'll fire for the first or the second. And he said, okay, well, maybe this is because the supplementary eye field keeps track of sequencing, which is what they were saying, or, that monkey's about to get his reward at the end of those three. Maybe it's just about reward. And he thought it was more likely to be the latter. And so he said, well, let's set up an experiment to try to, check on that. And what he did was, what he and the graduate student did was they did an experiment where they had two different kinds of experimental manipulations. One was the same sort of three-object serial visual saccade task, that's eye movement task, and they looked at the difference in firing rate for the third object versus the first, normalized here. And then they also instead manipulated the reward size, either big or small, and they did the analogous thing for reward. Looked at the firing rate for the large reward versus the small. And I was there when she reported the results from analyzing 54 neurons, and she said, well, the correlation is 0.49, and he said, oh, really, that's smaller than I would have thought, because he thought it was really reward that was driving things. He thought it would be a substantially higher correlation, and I said, oh, well, these are noisy measurements. I mean, the attenuation is probably, the correlation is probably attenuated. Statisticians know all about how correlation is attenuated due to noise. And he said, oh, really? And I said, yeah, not only that, you can correct for it. It's been known for over 100 years. It's very simple math that shows, when you add noise to two variables, the correlation goes down, and it's very intuitive. Again, this is something that can be explained to pretty much anybody. If two things tend to go up and down together, now you add noise, they don't go up and down together as much. Okay, so Spearman had a correction. We started from scratch and did something really, this is the thing that was really simple. I'm not going to show the details, but we just used a random effects model. also called hierarchical Bayesian estimation. And so the idea is that the measured X and Y involves signal and within neuron noise. And then there's also neuron-to-neuron variation in the signal, and that's really what we want to capture with the correlation of. So just to be a little more specific, we assume each of these two measurements for a given neuron results from signal and noise, and we want the correlation of the signals, not the noise. So the simple model would look like this. So we want the correlation of these guys across neurons. We can estimate the correlation because we have information about the magnitude of the noise. This is all done in repeated trials, which is to say repetitions of the same experiment over and over again, typically something like 15 or 20 times, and that gives us the noise magnitude that we need. So here's some simulation results from the simple analysis that we did. We look at the mean squared error from the uncorrected correlation with 15 trials. Here's the mean squared error here. If you use Spearman's correction, it's much smaller than the uncorrected, which is good. But if you do it the way we did, it's actually much smaller than Spearman's method. And if you look at the 95% interval, confidence interval coverage probability, if you fail to, this is a Z-transformed correlation. If you don't do the correction, terrible coverage. If you do, by the way, this method was proposed in only a couple of years before we published our paper in the psychology literature. They said, take the ordinary Fisher-transform confidence interval and then shift it by Spearman's correction, and that's what this is. And you get pretty close to the right coverage, but actually the Bayesian method is almost dead on. This is just illustrating the kind of thing we do all the time in statistics, and my point really is this is an elementary problem. It's pretty easy to explain. So here are the actual data results. The correlation came up quite a bit. Here's the confidence interval, fine. So the point that I make to the students is that by taking account of uncertainty, we improve the estimate. So statisticians focus on uncertainty all the time, but it's not always about uncertainty per se. It's also about improving estimation. And furthermore, as I was saying all along, we can exemplify the statistical paradigm here without formal details. And a last point is, this is really useful, I think, this particular example, attenuation of correlation, is really useful for discussing a really important point that we, I think, often make, but we certainly should make in talking about regression, which is about conditioning on variables in order to make an adjustment. One of the things people do, especially in social science, but in lots of domains all the time. And the point here is simply that if you condition on a variable and it's not really the variable you'd like to be conditioning on, or there's so-called errors in variable measurement and so forth, the effect of that conditioning is diminished, it's attenuated. And therefore, you're not really adjusting for what you think you might be adjusting for. Again, fundamental point, and just going through this kind of simple example helps to bring that out. Again, what I'm really advocating is that all of this should be done in the most basic courses and in our textbooks, such as the one Emery and I have written. Okay, a more involved example, sort of more interesting statistically, involves neural synchrony, and I can only just kind of run through this briefly, but I thought it was worth doing. The problem here is to try to look at the synchrony among two neurons, and it could be more than two, but let's take two neurons, which means that they both fire within the same time bin. Here, the time bin that we were using was 5 milliseconds. And that this would occur more than you would expect by chance. And again, the data I'm going to show you in a second come from the same array, slightly different experiment, but actually the same monkey and the same guy collecting the data. This is actually a subtle phenomenon, but you might wonder in neurophysiology why someone's interested in synchrony. And I will just say, I could go through this at some length, but I would just say it's been proposed to be really relevant for certain phenomena such as visual attention and generally communication across areas in the brain. It's very contentious. My view is really, I'm fairly agnostic about, I'm interested in whether it really occurs and is important, but to my way of thinking, regardless of the outcome of that debate, there's a feature of the data that we ought to be able to explain, and to do that, we devised a methodology based on point process regression. It is a subtle phenomenon, however, and I'll give you some indication of why it's subtle in a second. So the basic starting point is point processes. So here, a statistical model of a spike train involves two things: a probability density for the spike train in terms of an intensity function which will represent the firing rate for that neuron. It's a simple universal formula, it always applies. And then secondly, you need a model for the intensity. And this is something that Emery in particular pushed from the very beginning, and I quickly followed. And we are both effectively following what David Brillinger said in the statistics literature a few years before that. Actually, so once we have this, the point for us really is that we can apply quite a battery of existing statistical methods to this problem once we've put it in this form, because we have all of generalized linear model technology, modern regression technology to throw at this problem. And I should again repeat, this intensity function is going to represent this fundamental thing, the firing rate. And x here is just standing in as a vector. It's anything that could be driving this neuron to fire, that's anything that's been measured. And it could range in dimensionality from just a few dimensions up to many thousands of dimensions, depending on the problems. We've worked problems of both types. Just to say one more quick thing, it's really kind of an aside. When I said that we get the data in discrete form, and we typically actually analyze in discrete form most of the time, but we think of it in continuous, as a continuous point process, there's this fundamental phenomenon that, in continuous or discrete time, we can write the probability of a spike train, any old spike train. We get a likelihood function, and the two representations are consistent in the sense that if the bin size goes to 0, we get the likelihood functions are going to be essentially the same. So here, one way of expressing this is, indicates what's going to be happening here is the probability of a spike in a certain time bin given these variables is approximately the intensity times the bin size. So just to do the synchrony problem, I just wanted to mention, and I can't go into the details, but I found it very interesting. We had to solve a statistical problem, which is how can we have a mathematical framework according to which we can go back and forth between discrete and continuous time while allowing for synchronous spiking. The problem is the regularity conditions for point processes say no such thing as two simultaneous events. How do you get around that? How do you get likelihood functions that say, okay, it's almost the same thing in discrete time as continuous time? This took me many years to come to just coming back to this problem over and over again. I was very puzzled by it. We ended up with a very simple way of doing this, but I'm not going to go into it, I'm sorry to say. I think it's a reasonable justification for what we wanted to do anyway. So here's just an example. Here's the question. I mentioned the animal was anesthetized, and I'm going to show you the effects of this in one of these pictures in the slide. One of the things that happens under anesthesia is you get these what are called slow wave oscillations, and I'll show you this in a second. So a question that's going to come up is, to what extent is excess synchrony, meaning statistically significant synchrony, above chance due to the oscillation? So here, the pictures are going to illustrate this. So first, let me show you one neuron spike trains across 125 repeated trials, as nearly as possible, exactly the same experiment. There's some variation always in the timing of these spikes. And this is actually a grading, just like I showed you at the beginning, a bar of light moving across a receptive field, but it's moving always in very different orientations. Every 300 milliseconds, the orientation is shifting. And so when it's in certain orientations, you see high firing rates for this neuron. What's shown down below is a summation of all the spikes here, normalized so you get spikes per second. That's a very common diagram that people use. And here's the same diagram for a different neuron showing somewhat different response characteristics. But again, on the y-axis is different trials for that one neuron. Over here, what Ryan did is he did a different diagram where on the y-axis, it's not different trials. This is all the first trial, but on the y-axis, we've got different neurons. So here, there's 128 different neurons, all in the first trial, and then all in the second trial, all in the third trial, and so forth. And what you see is bands of activity that seem to be running across almost all of the network, and they are, every second or two. This is time in seconds. These are the slow wave oscillations, and as you go from trial to trial, they occur at different times. So that's not stimulus lock. The stimulus is exactly the same on every trial. It's why the spikes line up here according to the stimulus. They don't line up across trials here. But the problem is, if you want to know, is the stimulus driving synchronous spikes? Well, you're going to get tons of synchronous spikes here just because of the anesthetic effect. We ought to correct for that. And in fact, this guy, Ryan Kelly, the main point of his thesis was to go through various analyses and show how to correct for that phenomenon. So here's the output of our results, just illustrated in 2 pairs of neurons. The first pair is this top-bottom. You've got over 1,000 different spikes across trials here, and the circles indicate the times at which both neurons are firing within the same time bin. You'll see the circles line up top to bottom. And here's a different pair of neurons. Again, these circles line up top to bottom, indicating when these 2 neurons fired within the same time bin. And for both pairs, there's highly statistically significant excess synchrony. But we don't know yet whether it's caused by the slow wave oscillations or whether it's actually beyond that and maybe then presumably due to the stimulus. So it turns out we introduce a covariate for slow wave activity, the network activity, and then the left pair, once you introduce this covariate, it does seem that the synchrony was due to the slow waves in the sense that the synchrony here is no longer statistically significant. On the other hand, and by the way, if you look at the coefficient in the standard error, it's very close to the value in terms of the, standard error is small, the value of the coefficient is very close to the null value. And here in the right pair, on the other hand, this remains highly significant even with the coefficient, even with the slow wave covariate. That's the kind of thing that we can do. But immediately a statistical issue comes up, and people, when I present this, often people will interrupt me and start asking about this. Didn't you have 128 neurons, and then don't you have thousands of combinations? And yes, we have 8,000 pairs, so don't we have to worry about false discoveries? Absolutely. So a very common problem, a statistical problem in neuroscience is we want to infer connectivity. It could be synchrony, it could be something else entirely, among lots of neural signals based on, it could be spike trains, fMRI, EEG, MEG, and we're always, or we're typically trying to test many null hypotheses at thousands of nodes in a graph. So again, we have to protect against false discoveries, and I want to just quickly review one approach to this, Bayesian protection against false discoveries, which has been discussed in many places, and its connections with false discovery rate has been discussed also by multiple authors, and there's a couple of nice papers by Efron on this that I wanted to cite here. And then we've got a paper that I think has not come out yet, but it's about to come out in JASA, and this is, again, a very small extension of this methodology, I'll indicate. So it starts with Bayes' theorem. Probability of a hypothesis given data is written in terms of the probability of the data given the hypothesis. Null, we have null here. We'd also need the alternative. Here's the posterior probability of the null is what that notation means. And if you pick values for this P of H0, so-called prior probability of the hypothesis, if you fix a value for that and the probability of the alternative, it's equivalent to look at the Bayes factor here, which goes back to Jeffreys in 1939. And I wrote a paper with Adrian Raftery in 1995 in which we just tried to ask, in what ways does Jeffreys' concept apply with modern technology, modern computational methods? How does it play out in real examples? We had 5 real data examples in this paper. And excuse me for saying this, but it introduces an irony here. This turned out to be the most highly cited paper in the mathematical sciences over a 10-year period, much to my surprise. Here's the irony. I've almost never used this method. I've used it a few times. The reason that I don't actually like it that much, I like it as conceptually, but it's hard to apply in practice, is that this probability, the probability of the data given the alternative, can be hard to specify. A little more specifically, you need a prior probability, typically on some parameters, under the alternative hypothesis, and that matters. The answers change as you change that prior distribution, and in a lot of cases, it's pretty arbitrary, and I'm not comfortable making scientific conclusions when there's something arbitrary going on in the statistical modeling. Okay, so this is a wonderful method, but it requires that we know these distributions, the null and the alternative distributions. The null is often not that big a problem. It's a sort of a standard statistical null, although In some cases, it may be good to try to estimate that from the data, as we're about to do. It's the alternative that causes the biggest headache, typically. But network data give us a great opportunity because we can actually estimate these distributions from this rich network data. So this goes back to this Bayesian version of false discovery rate. We assume each test statistic at each node follows a null distribution with probability pi, an alternative distribution with probability 1 minus pi, and we can use the network data to estimate both of the distributions, the null distribution and the alternative distribution, non-parametrically. And we can then incorporate covariates to improve this. This is our contribution. And in the case of synchrony, we know that if neurons are close together, they're more likely to be synchronous. And so we can actually, we know how far the electrodes are apart. We can incorporate the distance as a covariate. This requires quite a bit of care. The first author, James Scott, is a very good, relatively young professor at University of Texas, Austin, if you don't know him. And it was really mostly his work. I was kind of the person who brought the problem and talked to him about it. I thought he did a very good job with this. It's very careful, and I think it's interesting. The simulation results he did show that it's a reasonable method, reasonably robust. And the interesting thing, of course, is that as you add information, you get a lot more discoveries controlling at the same false discovery rate than vanilla FDR. And this just illustrates this. Here's distance, and in gray are all the discoveries you would get by either method, and in blue, the extra discoveries you get by adding the covariate here, which are at small distances. As those are the places you would expect to get more discoveries because you've injected that information. And the covariate effect here through essentially a logistic regression model is actually very strong for distance. So that was all nice, but I wasn't completely satisfied. These procedures are still ultimately screening methods. What we really want to do, screening in the sense that, it's hard to be very confident about any of the conclusions that come out of it, I think. What we really want to do is replicate. So this relates to something that, Andrew was talking about too. There's this so-called crisis in reproducibility. It's been discussed in many places. Here, there's these articles in Nature. Some of it's linked to statistics. There are statistical errors discussed. I think one of these is actually by one of the authors, or it refers to one of the authors that Andrew showed. And in fact, it's gotten pretty extreme. If you haven't heard about this, that one of the journals bans p-values, kind of throwing the baby out with the bathwater. But one of the points that some people make is that part of the issue is the p-value. There are many issues here, and Andrew talked about some of these. But one of them is the p-value fallacy, the idea that p-values are often interpreted as the probability the null hypothesis is true. And some people have suggested that we should instead use Bayes' theorem and Bayes' factors to solve this problem, to avoid this problem. And as I've indicated, that's not my view. I don't think that by itself is going to solve any problem. And again, it's because now you're faced with yet another problem. How do you specify that probability? But in thinking about this, it seems to me that this could be not so much telling us something about statistics and the problems with statistics, but rather telling us something about science and the problems with science. So, if this is what we'd like to do, maybe we should change the science so that we can do it. In other words, we should repeat. So there's a truism. We believe the scientific results that are repeated across animals, across sets of subjects, across experiments. And I want to just go run through an example from my own personal experience in neuroscience. The one nice case where we actually did this because we were worried. Well, this involves directional tuning of neurons in primary motor cortex, a phenomenon that was discovered in the early 1980s, and the guy who discovered it had a postdoc named Andy Schwartz, who ended up moving to Pittsburgh and became one of my main collaborators some time ago. So the idea is when a monkey sits at a table and moves his arm in one of 8 different directions, shown here, and you record one neuron in primary motor cortex, the part of the cortex that is driving muscle activity, you get this, what was then a very surprising finding, which is that if you look at the neuron's activity when the direction is when you're in one direction of movement versus another direction of movement, in this case the neuron's firing much more in this direction than in this direction, which is very surprising because it's this abstract concept, and you're doing almost the same muscle movements in both cases. Somehow primary motor cortex was actually coding for the direction of movement or perhaps the velocity of movement. You can fit, actually, as it turns out, a curve to this pretty easily, a cosine curve. I'm almost done. And the point at which the neuron fires the most is called the preferred direction. And they then showed that you could combine these preferred directions by weighting by the firing rate and get what they call the population vector. Here's a representation of a whole bunch of neurons with different preferred directions, and the length of each is the firing rate, and the result of adding those together is the population vector, and they showed that actually points in the direction of movement. So this was very, very interesting. Soon after, they realized that you could, in principle, you ought to be able to create a prosthetic device. So you can combine the activity of dozens of neurons and then predict the movement that someone would like to do and actually have a robotic device. Here, the signal's coming out of the brain, and there's a controller somehow, and you can move a robot. I'm just going to show you a movie of a woman who was terribly paralyzed not too long ago, and she learned to think about moving her hand and could control a robot arm as a result. This is called BMI, brain-machine interface. She's going to mention BMI in a second. She's controlling actually a somewhat complicated robotic arm. It's got 10 degrees of freedom in this case. And this was the big moment when she could actually feed herself.
There we go.
One small nibble for a woman, one giant bite for BCI.
Brain-computer interface. So we realized that we could actually do something where we could use the statistical methods to learn about motor cortex, and the idea was we would take the algorithm and rotate around the field and force the animal, in this case, to learn a new transformation of what would happen. So the cursor is suddenly going to go in the wrong. If it's a cursor on a screen that he's controlling, it's going to go in the wrong direction. He would have to adapt to that, and there would be. And the idea was, let's see what's happening in the brain when he's adapting. And this was a 3D reaching experiment we did. But the postdoc in the lab, I wrote a grant on this, and I thought it was a great idea. The grant got funded. We agreed to do it. The postdoc had a slightly different idea. She said, oh, instead of that, we're going to rotate the preferred directions of half the neurons, which will have the same effect from the monkey's point of view of rotating the whole visual field, but half of the neurons will be rotated and half will stay the same. And she came back and said, oh, when we do that, the neurons that were rotated have their preferred directions shifted, but the neurons that didn't rotate, that we didn't manipulate, don't. And I said, what, are you kidding me? You mean the monkey has no knowledge of what you did with your algorithm, right? All the monkey's seeing is the cursor's rotated. And you're saying that the brain has solved the credit assignment problem. There's going to be thousands of neurons in this network, and the brain has figured out which neurons you manipulated? No. And Andy Schwartz, the PI of the lab, he said, no, I don't know. But she kept working at it, coming back. We started to get convinced maybe there's something to it. And in the end, what we decided, she was about to leave after a lot of work, and she was really upset we weren't letting her publish this. And we said, okay, do a third monkey. She had already done on 2 monkeys. Do a third monkey. You've done all your fooling around. Repeat on a third monkey. She did that. One of the reasons, before I get to that it was worrisome is here's the nature of the effect. The non-rotated, this is a representation of preferred directions, but the non-rotated ones are in blue. Here's the mean. The rotated ones are in red. It's a very tiny effect, but highly statistically significant. All the red flags are going off. Bias, 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 right? Okay. Run a third monkey, see if the same thing happens. Sure enough, it did. We published the paper. But I was still a little bit worried, even though I was in PNAS. Okay. So then the new postdoc came in, and I said, okay, let's run the new experiment differently. Instead of a 3D experiment, let's do a 2D experiment on a screen, which is actually controlled a little better, and let's see what happens, and get even more data. And there, the same thing happened, and then I kind of believed it. And I want to tell you, the point now, from what I was saying before, is we want to replicate And actually, the first experiment can form a probability distribution for the second experiment. What I'd like to see is not an exact replication, but a replication that's a little bit different, but enough to get a prior distribution. And when we haven't done the Bayesian calculation here, but it turns out if sweeping aside some details, roughly speaking, the 2D result is about 5 standard errors from the null and about 1 standard error from the result in 3D. So I think when we do the Bayesian calculation, it's going to be overwhelming odds that it was a real replication. Now, there could be some hidden bias in both of these experiments. It's possible, but at least it seems like a strong replication. And ideally, we'd really like to do this across labs, not just across different kinds of experiments. Okay, so in summary, statistical thinking involves statistical models and evaluation of procedures. I showed you an elementary example involving attenuation of correlation, an more advanced example involving neurosynchrony and FDRR, false discovery rate regression. I also wanted, I showed you, I said, we ought to be replicating more often. And this would, in principle, allow us to use Bayes factors. Now, I don't want to suggest that we ought to be doing Bayes factors. That's not my point. I don't really care that much whether we do Bayes factors per se. To me, although I like Bayesian methods, the more fundamental thing that Bayes usually has done for us, and it's done it historically, is to bring to stark relief an essential nature of the problem and help us understand it better. And that's the spirit in which I'm saying I think the Bayes factor formulation helps us see what we need to do and the way we need to get information in replicating scientific experiments. Thank you.
We have time for 1 or 2 quick questions.
I think replication is great, but I've never actually done a pre-registered replication in my life. There's one I've been wanting to do for a few years. I keep throwing students at it, and it's like too hard for them to replicate the original study. And part of that is in social science, we often have these data sets that are harder to get. I can't just stick a few more monkeys in the lab. I need to do a new national survey or run it.
Please don't do this. We're talking about potentially a couple years worth of work. I mean, these are not necessarily so easy. I think the neurophysiologists would differ with you on that.
So maybe the message is I should work harder to do 1 for 2.
Maybe that is the message. And by the way, I really want to emphasize my own personal bias is you don't want to do an exact replicate. You want to do a replicate that's a little bit different because I think you have a greater chance of avoiding bias. Yeah.
Oops. Sorry. Rob, I presume all of this is testing means, right? Essentially, mean directions.
The thing that I was just showing? Yeah, essentially
Okay,
differences.
So what would happen if you looked at actually the distribution of spikes, let's say variances or some other features, whether that might change substantially too? I mean, it depends what test
right.
you do also.
Right. Well, I think the reality is I don't know, and I don't know that we're going to find out because I think at this point people have moved on. But someday somebody may come back to this and try to answer that. So it's a good question.
I think there's one last question over here.
Yeah, I wanted to, so I'm certainly on board with that we need to have more replications. I like replications a lot. Something that came up in Andrew's question, I guess in your students' concerns, is that the incentives tend to work the other direction.
Yeah.
And so is there a way of us that we can set up the incentives such that it would be in people's advantage? I mean, it's obviously in the advantage of us as a group to do replications, but it's not necessarily in anyone's individual interest.
Yeah, it's a huge problem. People are starting to try to do that. I know there are groups that are trying to do it. There are labs that have started to collaborate because they recognize how important it is. And ultimately, I think that for the lab-based collaboration, I think they realize that the science will only be better, and they're still going to get to publish their paper in Nature, but it'll just be better. And the thing that I know some people have done, there's journals that have started now, and I think it's in psychology, where they're devoted to replication. In fact, Andrew showed a slide from some journal, I think that was specifically about replication. So, there are efforts underway. I think there are things that can be done. Tenure committees have to agree that those are valid publications, right? And letter writers, actually, have to agree. And so it's a bit of an uphill battle on this, but we all have to push for it.
Great. Hopefully we can continue the remaining questions with the discussion, but now we're going to break for lunch, which I believe is in the atrium. Let's thank our speaker again.